Hello, everyone. Welcome back to my video tutorial for TCR sequencing data analysis. Today I'm going to demonstrate how to analyze the gene usage. So you know from the TCR sequencing data, we have the nucleotide sequence information for the VDJ gene for both TRA chain and the TRB chain. We can use the get array immune receptor repertoire context function together with vCore, jCore, or dCore to get the gene information for the V gene, J gene, or D gene. So first we can call the V gene, then use the group abundance function to look at the top 20 V genes in the TRA chain. You can see we use the vCore here. Then in the group by function, we use the VJ1 vCore because the TRA only has V and J segment. So in this code, if we run this, we will see TRA V gene. Let's run. You can see we generated a bar plot to show the top 20 TRA V gene in this data set. Because we set the normalize as true, so you can see the y-axis is the fraction of cells in the cell clusters, and the x-axis show the top 20 TRV gene name. The color in the bar plot represent the cell types in this data set. So we look at the V gene in the TRA chain. Then we can also set the core as J core. Then look at the J gene in the TRV chain. Again, we are going to look at the top 20 TRA J gene. If we run, you can see we generate a bar plot to show the top 20 J gene in the TRA chain. So in TRA, we only have V and J gene. But in TRB, we have the V, D, and J gene. First, we can have a look at the V gene in the TRB chain. Again, we can use the vCore to look at the V gene. But here you can see in the group by function, now we set it as VDJ. Then you know we are calling the V gene in the TRB chain. So here is the vCore. Let's run it again. You can see we generated the bar plot to show the top 20 V gene in the TRB chain. So next we can look at the D gene. Then we need to use the dCore, and also the group by function will be VDJ dCore. Let's run. You can see in the TRB chain, we only have D1 and D2 gene. And again, the color show the TRB D1 and D2 gene usage in different cell clusters. So far we use the VDJ core to see the top 20 V and J gene usage in the cell clusters and also the D1 and D2 gene usage in the cell clusters. If you want, you can also look at the specific clone types for their usage in the cell clusters. For example, here we can select 5V gene. You can see here the V gene names in the TRB receptor to look at their usage in the cell clusters. Because we are going to look at the V gene, then we need to use the vCore, and the group by name will be VDJ1 vCore. If we run this, you will see the gene usage that we selected by their gene names. Now you can see the color represent the gene names we selected here. Then the x-axis show the cell clusters. You can see the percentage for the usage of selected TRBV genes in the cell cluster. So I just use the TRBV gene for the demonstration. You can use the same code here to promote other genes, for example, the D genes in the TRB chain, the J genes in the TRB chain, and also V and J genes in the TRA chain. So far, we only look at the usage of individual VDJ genes in the cell clusters. We can use the scTCR plot to look at the combination of different gene usage in the cells. Let's run the plot first, then we can explain the result. Let's run. You can see we generated the plot. Now you can see the y-axis is the clone IDs, then the axis show the genes. You can see the first one is the V gene in the TRA chain. Second one is the J gene in the TRA chain. Then here are the V gene, D gene, and the J gene in the TRB chain. So in this figure, you can check the combination for the TRA chain and the TRB chain in individual columns. In this figure, we plotted the order columns. You can also select the individual columns to plot the gene combination. For example, we can use the clone ID 68, 101, 127, and 161 to see the gene combination in those clones. We can run it again. You can see now we have the gene combination VJ gene in the TRA chain, VDJ gene in the TRB chain. For the 4 clone types that we selected here, so you can see from here the gene combination for the individual clone types. So I showed you how to use the V-core, D-core, and J-core to look at the gene usage in different cell clusters. In the TCR sequencing data, we also have the amino acid sequence for the CDR3 region. So we can also use the spectral type plot to look at the amino acid length distribution in different cells. Again, we can run to generate the plot first. You can see we generated the plot. Now the x-axis is the amino acid length, and the y-axis is the number of cells that have the same amino acid length, and the color represents the cell clusters. So you can see the CDR3 region with 13 amino acid sequence has the highest frequency in this dataset. The next one is the 14 amino acid length. You can see most clones have the amino acid between 11 to 16 in length. So here we plotted the amino acid length for all the clone types in different cell types. You can also select individual clone types to look at the distribution of the amino acid sequence. For example, we can select 5 CDR3 sequence that contains the amino acid sequence for TRBV genes. If we run the plot, you can see we generated the amino acid length for 5 selected V gene sequence. And the x-axis show the total amino acid length. Then the y-axis show the fraction of cells in the samples. So we use the bar plot to show the amino acid sequence length. If you like, you can also use the Rigi plot. We already made the same plot, use the bar plot showed here for the amino acid sequence length. Then we can use the same data to make the Rigi plot. If we run, You can see we generated a rigid plot to show the amino acid sequence. You can see the x-axis is the length of amino acid sequence, but now in the rigid plot, the color represents different cell types. So that's today's demonstration. I showed you how to look at the gene usage for TCR sequencing data. We made the plot for both the nucleotide and the amino acid sequence. So I'm going to stop from here for today's demonstration. I hope to see you in my next demonstration.